These are the goals for the photosynthesis chapter. By the time you've completed this video, you should have all the information you need to achieve these goals. Here's the chemical equation for photosynthesis. Take a moment to pause the video and ask yourself what the reactants are. What about the products? The next two slides review common leaf anatomy. Perhaps some of the most important features are the cuticle and the stomata. The cuticle is a waxy covering that prevents moisture loss and the loss of excess carbon dioxide and oxygen. The stomata are pores in the epidermis of leaves that open to allow for gas exchange and close to prevent moisture loss. Here is a full-color depiction of the leaf cross-section model as shown in your manual. All of the tiny little green circles indicate individual chloroplasts. Remember the typical anatomy of a plant cell. The processes of photosynthesis happen in each chloroplast inside the plant cells in the green parts of plants. The next two slides review the structure and function of the parts of the chloroplast. Please pause the video if you need to review these structures. This slide introduces us to the next activity examining photosynthetic pigments. This is one of our goals for this chapter, so be sure that you take lots of notes regarding this procedure. In this activity, we separate four different photosynthetic pigments: chlorophyll A, chlorophyll B, carotene, and xanthophyll. As you can see from this diagram, energy increases as the wavelength of light energy decreases. This absorption spectrum diagram shows the four different photosynthetic pigments and at which wavelengths they absorb the most energy. As you can see, chlorophyll A is a blue-green color as compared to chlorophyll B, which is a yellow-green. Xanthophyll is bright yellow, while carotene is orange-yellow. You can follow along with this procedure in your manual. Paper chromatography is used to separate the four pigments from any leaf. Chromatography paper is highly hydrophilic and will strongly bond with polar and hydrophilic pigments. For this lab we use spinach leaves, though any plant leaves could be used. The leaf is laid over the pencil line on top of the chromatography paper. A quarter is then rolled back and forth over the pencil line at least 15 times to transfer a heavy solid line of the spinach pigment onto the chromatography paper. Your instructor would then add a small amount of the chromatography solvent to a large chromatography test tube. The chromatography solvent is hydrophobic. Once the solvent is in the tube, the chromatography paper strip is inserted into the tube, making sure the solvent touches the paper but does not cover the pigment line. Then a cork is used to close the chromatography tube, and then we wait for 10 to 15 minutes. The chromatography solvent will diffuse up the paper, coming into contact with the pigment line. As the solvent continues to move up the paper, it will carry the four pigments with it. The pigments will separate on the chromatography paper based upon their differences in polarity. Once the solvent has diffused to within 2 centimeters of the top of the paper, we remove it to dry and analyze our results. Here are two pictures which illustrate how the four pigments separate. You can see the chlorophyll B stays close to the original pigment line, meaning it travels the least far. In contrast, the carotene travels the farthest up the paper. In the next activity, we examine the requirements for photosynthesis. In which organelle does photosynthesis occur? The chloroplasts. Look at the photo of the leaves shown. Which parts of the leaves contain chlorophyll, the green parts or the white parts? Follow along with this procedure in your manual. There's a place for you to draw a coleus leaf. You can use one of the leaves shown in the photo on the previous slide. The first step is to drop the leaf into a beaker of boiling water for 3 to 5 minutes. This process ruptures the plant cell walls and disrupts the membranes within the plant cell. Next, we place the boiled leaf into a smaller beaker containing about 30 milliliters of ethanol. This smaller beaker is then placed back into the larger beaker of boiling water. The leaf is now boiled in the ethanol for 3 minutes. During this process, the chlorophyll is extracted from the leaf. Once complete, the leaf will be virtually colorless. Now the leaf is placed into a petri dish and covered with iodine. The iodine will react with any starch that is present in the leaf. The iodine and starch interact to form the distinctive blue-black color change we've seen in lab before. In which parts of the leaf do you think we'll see this reaction? As you can see, the blue-black color change takes place everywhere the leaf was green, meaning the parts of the leaf that contain chloroplasts. This is the end of our procedures for the photosynthesis chapter. Now be sure to go through all pre-lab questions, post-lab questions, as well as lab activity questions in the chapter to be sure you understand the procedures.